And so you want it to not be toxic to basically anything except for the pathogen. I would say that's probably the thing that most people aren't aware of, is that balance between making it both very effective against the pathogen but also not harmful to anything else is quite tricky.
Hello, everyone. Welcome to the Vandal Theory, a podcast about science and research at University of Idaho. My name is Lee Cooper, and I'll be your host for today. I'm an editor and science writer here at the university, and I am joined by my co-host. Hi, Danae.
Hi, Lee. I'm Danae Lenz, and I'm the External Communications Coordinator at U of I.
We're back for season 11. Can you believe it?
I honestly can't. I'm really excited. We got some good ones this season.
Absolutely. So today, we're going to talk about crops and garden pests, specifically fungi. And to get us into this, Danae, you're a gardener, yeah?
I am indeed.
Okay. So how do you keep pests out of your garden?
I won't lie. Most of it is just planting things in the ground and crossing my fingers and saying a prayer. But one thing I do that I think is kind of cool is there's flowers that you can plant that deter pests. And so that keeps it entirely organic. Marigolds, petunias, lavender, they all do sort of different things and keep different kinds of pests away. But that is something that I do. And yeah, it livens up my tomato garden.
Makes it look just a little bit prettier as well.
Mm-hmm.
I am not a huge gardener. I have a small herb garden, and really, I'm a terrible plant parent. You know, once I put them in the ground, they're off on their own. I had a frog come visit this year for like most of the summer. Maybe he ate some bugs. I don't know. Yeah, that's about it.
Yeah, we're basically on the same page there.
Yeah. So today we're going to meet Marty Ytreberg, a professor in the physics department at University of Idaho and the director for the Institute for Modeling, Collaboration, and Innovation. Alongside a large team of other interdisciplinary scientists, Ytreberg is designing brand new fungicides to protect Idaho potatoes, and someday, hopefully, other crops as well. He's going to delve into the steps his team uses to find fungi's weak spots and what makes a successful fungicide. Ready to go?
Let's dig in.
Marty, welcome to the Vandal Theory.
Thank you.
It's so good to have you on today. Do you want to introduce yourself to everyone real quick?
Sure. I am Marty Ytreberg. I'm a professor in the physics department at University of Idaho, where I've been almost 20 years now. It'll be 20 years this fall. And I'm also the director of the Institute for Modeling, Collaboration, and Innovation, otherwise known as IMCI.
And what does IMCI do for the most part?
The primary thing that we try to do is to bring kind of model-based collaborative research to the University of Idaho. A lot of it is biomedical focused, but we're much broader than that. So pretty much anything that involves modeling broadly defined, we like to be involved in.
Which brings us to what we're going to talk about today.
Yes.
You've started a new venture looking at fungicides, or the chemicals that take out fungi. The way you started this project has been a little, you took the winding road to get there.
You could say that.
Can you tell me how you started this project and maybe getting towards what are you actually testing now?
Yeah, this all started, I think it was about 3 years ago now, where a group of scientists, including myself, got together. And essentially, we were all interested in the idea of designing therapeutic molecules. So some of us, such as myself, do molecular modeling, where we look at things like proteins and how molecules might interact with proteins. We had other people that were part of that group that were interested in essentially making the molecules, say from the chemistry department. We had other people who did experimental testing. And so we just kind of got together and were kind of brainstorming what could we work on. And of course, with a group of people like that, the first thing that comes to our minds is kind of human therapeutics.
Of course.
But we quickly thought to ourselves, well, we're at a small research university in Idaho. We're not going to be able to compete with the billion-dollar, mega pharmaceutical industry. And so things really took a turn for the better in our discussions when we invited Brenda Schroeder from the College of Agriculture and Life Sciences to join us. And Brenda has expertise in plant pathology. So she quickly kind of turned us on to this idea that potatoes, which of course are an important crop for the state of Idaho, have all these fungal pathogens for which there are not a lot of ways to control them.
Loved by all organisms,
Yes.
not just humans.
Yes. And so what we realized is not only did we have kind of the right kind of skill sets to be able to start thinking about designing fungicides for potatoes, but that also, unlike drug design for humans, we actually have the ability to test them on plants. I will say that one of the things we also realized is that the methods that we're using, which I'm not going to go into a lot of detail on, but they are unique. So in that sense, we're not directly competing with what the big agricultural companies are actually doing. Our method for designing fungicides for potatoes is actually quite different. So that's kind of how we arrived at this idea of designing compounds that could be fungicides for potatoes.
All right. Obviously, anytime you have a pest that's going after an agricultural crop, that's not great for the farmer and will hurt their bottom line. We have some fungicides. Fungicides?
I say fungicides. I think there's no wrong way to say it necessarily. So either works.
Fungicides for potatoes and for other crops. You're designing almost a whole new class. Yes? What does that mean? Put that in context for me.
Yeah, so fungicides all fall into certain classes kind of based on how they stop the fungal infection. People in agriculture or in this world of fungicide design would call it the mode of action of the fungicide. So the way that it's operating on the fungal pathogen to essentially stop that infection. And there are limited numbers. Somewhere, depending on kind of who you talk to, somewhere around maybe 15 or 20 different kind of classes of fungicides are out there in agriculture right now. And you might think, well, that's enough, right? The issue is that what happens is that you can develop resistance to these fungicides in the same way that certain types of bacteria might become resistant to, say, penicillin or some other sort of drug. And so because of that, whenever resistance pops up, what can happen is that certain classes of fungicide no longer become effective. You need new classes in order to be able to treat the fungal infection at that point. The other thing I'll point out is that there are certain types of fungal infections, for example, the causal fungus for what's called verticillium wilt.
Okay.
Which currently doesn't even have a chemical control, meaning that right now, if the farmers want to treat for this particular pathogen, they actually have to fumigate their crops, which is very expensive and time consuming. They don't have the ability to, say, just crop dust or something like that. And so there are certain pathogens out there that we're hoping to be able to find controls for where there are none.
Okay. So twofold. You want to make sure that they don't become resistant. And two, there are actually a few diseases and stuff that don't have any offset at this point.
And just to clarify on the resistance point, the reason for having new classes is really just so that we know the resistance is going to happen. And regardless of what compounds you design, nature will find a way around it, right? And so the idea is you want to have additional options. So when that resistance happens, you can switch to something else that is effective. And so the more options you have, the better.
Okay,
right?
Cool. That was very Jurassic Park moment. So can you broad overview? I know there's many steps to doing this, but broad overview, how do you start to look for a new fungicide?
Yeah, so essentially, we sort of start from the perspective of could we design a fungicide that has a new class? In other words, it has a different mode of action compared to the ones that are out there.
And we're talking like one mode of action might be me as a fungicide stops, step 3 of reproduction, while another one is I stop them from processing this nutrient that they have to live, or like,
Yeah, I
right?
can give you a specific
Oh,
example
okay.
if you'd
Yeah.
like. So a specific example, these are called QOI fungicides. The idea is that this particular class of fungicides, what they do is they bind to a particular protein
Hmm.
that is involved in the pathway that the fungus uses to create ATP. And I
Okay.
think most people understand that ATP is kind of the primary energy source for a cell. So if you shut down the production of ATP, what happens is that the cell can no longer reproduce, which means that the infection goes away. And so that's one example. And you can imagine that that pathway that I just talked about that produces ATP could involve multiple different proteins.
Sure.
It's a kind of a complicated network of events, right? And so if you can disrupt that chain reaction at any point, you can potentially shut down or limit the amount of ATP they can produce.
Take out a different domino from
Yeah,
that
exactly.
process.
And so but going back again to the resistance point, oftentimes nature is pretty clever, and the way that it evolves around that means that it eliminates all the things that you might have thought you could use to disrupt that same pathway. Hence the need for different modes of action or kind of chemical compounds that kind of stop different production pathways from happening.
Okay. So you're looking at the fungicides that are out there and which pathways they're blocking, and you're looking at kind of different parts of that to work on.
hmm
That's how you start it?
Yeah, and essentially what happens is that we have kind of a collaborative team that does a combination of molecular modeling, experiments where they're actually testing different chemical compounds in the lab, and chemists who design the molecules and can essentially tweak the molecules based on what we tell them to try them to make them more effective. And all these things kind of work together to try to find a chemical compound that actually does what we want it to do.
A decent amount of trial and error, it sounds like a little
Quite a bit
bit.
of trial and error, but also what I would say is informed trial and error.
Sure.
So it's not random. It's where we're trying to all communicate and use the knowledge that we have to try to make informed decisions about what to do next.
I've been on campus for 8 years now, and worked with quite a few different teams. Yours is especially interdisciplinary, to be able to do this, which seems right here is where that comes into play very nicely.
Yes, we have quite a few people on the team, and I would say that we have people that do kind of modeling. We have people that do experiments. We even have people that are engineers
Hmm.
with an eye toward if we do develop some of these compounds, what's the best way to actually use them in the field. So we kind of have, you could say, people to fit into every step of the pipeline that you need to develop an effective fungicide.
What are some of the questions that come up that maybe people wouldn't think about when you're designing a fungicide? What are some of the things you have to take into account?
Yeah, I think probably the number one thing is that most people would think that your job is done when you find a compound that kills the fungal infection. It's like, well, if it's effective at killing the fungus,
That's the baseline. You're over
that's
it.
that's you're done, right? Well, the truth is that's really just the starting point. You need an effective compound. You need something that shuts down that fungal infection. But that's really just the start of a very long process. And so if you want this to actually be a fungicide product that can make it to the market someday, there's a lot of other considerations. Of course, the most obvious consideration would be you don't want it to hurt the plant. So if it kills the fungus, but it also kills your potato plant,
You've really
That's not
not
a
gotten over that baseline.
exactly. That's not good. And so what you want is something that is very effective at killing the fungal infection, but is harmless to the plant. In addition, you have to think about the fact that this is going to be a chemical that is going to be sprayed in the environment.
Mhm.
So you have to ask questions like, how long this compound actually survive in the environment? Something that breaks down more readily would be better, right? Because
Mhm.
you don't want it ending up in our water supplies and so on. You also want to know, is it harmful to animals? Humans, of course, are the number one priority there, but other animals too, right? You don't want it to be harmful to birds or to other mammals that might come in contact with this chemical at some point. So a lot of it has to do with kind of toxicity
Mhm.
to other organisms. And so you want it to not be toxic to basically anything except for the pathogen. I would say that's probably the thing that most people aren't aware of is that balance between making it both very effective against the pathogen, but also not harmful to anything else is quite tricky.
So how do you look at that? Is that a actually you do tests, or is it, like you talked about, you're looking at this, pathway. Are you looking at pathways that don't exist in the other creatures, or how do you take that into account?
Yeah, that's a big part of what we're trying to do is we're trying to find essentially you described it quite well. Really, you're trying to find pathways that you can disrupt that don't exist in humans and plants. And so if you can find kind of novel pathways that occur in a pathogen, a fungal pathogen in this case, that don't occur in plants and animals, then it's less likely, it's not a guarantee, but it's less likely that you're going to do something that's harmful. And so that's kind of our starting point is that we're trying to identify pathways that are unique to the pathogen.
So how are you actually testing for some of the toxicity stuff?
The primary thing that we're testing is toxicity against the potato plant itself. And so the way that we're doing that is pretty straightforward. Essentially, what we're doing is we're taking these chemical compounds that are found to be effective at killing the pathogen, and we're essentially doing experiments in a greenhouse where you essentially you apply different concentrations of this compound to a potato leaf, and then you essentially just look to see whether or not it's harmful to the leaf. And so what you typically find is at very high concentrations, almost anything starts to become dangerous to the potato leaf.
Which is
But as
true
you,
for almost any compound for
right,
any organism.
right, exactly. And so the idea is you're trying to find compounds that the concentration that you would need to kill the pathogen is well below that threshold where it becomes dangerous to the potato plant. We also have plans, we have not done this yet, but we also have plans to test our chemical compounds in human cells. In other
Mhm.
words, just apply them directly to human cell lines to see if we can get a sense of toxicity toward human cells. But I will say this is not something that we plan to invest a lot of time and energy into. And the reason is because our goal is to find, if we can find a compound that's both highly effective, has a new mode of action that we can demonstrate, and that's not toxic to the potato plant, the next step would really be talking to someone who develops agricultural products, a company. And they would be the ones that would need to go through a fairly lengthy
There's a
process
process.
involving millions of dollars and approximately 10 years, believe it or not,
Wow.
before you can take a chemical compound and turn it into an agricultural product. Most people are surprised by the amount of money and the length of time. It's actually longer than for a human therapeutic.
Hmm.
And the reason for that, on average, it is. And the reason for that is because these compounds, if you're thinking about human therapeutics, you just have to prove that they're not harmful to humans. But for a fungicide product for crops, you need to show that it's not harmful to humans, other animals, plants, the environment, etc. So there's a lot more kind of regulatory hurdles that you need to go to that are quite time consuming and costly.
I mean, that makes sense logically. I
Yeah.
mean, Yeah, human drug would just be you personally.
Yeah.
So right now you're working on potato fungicides. Is this bigger than potato fungicides? Is this drop one in a very large bucket?
Yeah, we hope so. It is in the sense that the people, the expertise that we have in place is not dependent on potatoes and it's not dependent on a fungal pathogen. The method that we've developed, this pipeline that we've developed for potato fungicides, could just as easily be applied to fungal infections for other crops, for example, onion. And that's something that we've actually already started to think about. Other crops, rice, soybean, are more global crops that have quite a bit of issues with fungal infection. More locally, potatoes and onion are the big ones. So any crops, but then beyond fungal pathogens, you can also think about other types of pathogens. So this same technique that I was talking about with looking for kind of unique pathways that you can target, you could do that for a viral infection. You could do that for a bacterial infection. You could do that even for a nematode.
Okay.
And so really all the kind of major issues that are faced by crops could potentially be tackled with this sort of technique. Of course, it takes quite a bit of time to get to the point where you're actually doing that. But in principle, any of these other pathogens and any of these other crops could potentially use the same technique, the same pipeline.
Well, any last thoughts that you want to leave with the audience about what you guys are doing?
No, I don't think so. I'll just say that, we have a lot of plans for taking this approach that we're using and applying it to other crops and pathogens. And so we're really excited to build this kind of from the ground up, and we're hoping it continues to grow from here.
When it's wonderful, I sat in on one of your meetings. You have a decent amount of graduate students as well working on the project, and it's a pretty big team that will get a lot of experience out of this.
Yeah, we have 7 faculty members at the university, and each of us has, 1 or 2 graduate students that's working on this sort of stuff. In addition, we have some postdocs, we have some undergraduate students. So it really is a large collaborative effort. I couldn't even tell you off the top of my head how many people are involved, but certainly in the neighborhood of 20-25 people have been kind of touching this project at different points, which I love. I love to be part of an interdisciplinary team. So
Well, thank you, Marty, for coming in today.
thank you very much for having me. Appreciate it.
All right, Danae, what'd you hear?
Okay, so my biggest takeaway, the thing that blew my mind the most, was him saying that it takes so much longer for pesticides, fungicides to get approved for the market than human drugs. I've never really thought about it, but if you had just asked me, obviously it's human drugs, obviously. But then he explained it, and yeah, you just have to take into account the environment, other plants, animals, all these other things versus pharmaceuticals are just for humans. I actually went back and listened to that twice because it blew my mind so much.
I gotta say, I would agree with you. If you had just asked me, I would have totally gotten that question wrong. I would have said human drugs would take way more work and money. Yeah, that was my favorite part as well. One of the other things is I really like thinking about the problem-solving puzzle pieces of this, almost, where they're looking at however many steps it is for, in this case, he talked about how they create ATP, these cells. And they're looking at all these different steps and going, okay, that fungicide takes out step 3. We're going to go for step 5. Like the precision of that. And I mean, you're thinking about this from like a cellular scale, like the scale that this is working at is just mind-boggling to me.
Yeah, I also think I don't think he brought this up exactly, but I know from talking to other researchers that not all of the crop genomes are mapped. Like, in fact, there's not very many of them that are. So I imagine that makes it much harder to do the kind of work that they're doing.
Yeah, I would think so. And probably why they're trying to map more and more of them. Because, I mean, this is the kind of place where that's going to come in super helpful.
Yeah, the other thing I was thinking of, I guess it's not exactly pesticide related, but every year when once a year only, I decide to spray my lawn and kill the weeds. And when he was talking about, yeah, how do you just kill this thing that's like on the plant, but you don't kill the plant itself? And every year when I'm spraying my yard, I'm like, how do they just kill the weeds and not the flowers or the grass or anything else? And honestly, it's pretty incredible.
And now we know.
We sure do.
All right. Well, thank you everyone for listening to the Vandal Theory. You can visit our website, uidaho.edu/Vandal Theory, to learn more about our research. Email us at vandaltheory@uidaho.edu with any questions. And make sure to listen to us on all the podcasting platforms out there. There you can rate, review, and subscribe. My name is Lee Cooper.
And I'm Danae Lund.
Thanks for listening, everyone.